طيب بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. My name is Amjad Ajam, and today I will be discussing the third lecture of cell signaling. It's a short one and easy one, so I promise I will finish as soon as possible, inshallah. At the end, I added some practice questions, so please look at them because most of them are past questions, so that you know what things you have to concentrate on. And with that said, let's start. So my objectives for this lecture, first of all, we will discuss the structure of the steroid receptor. Then we will know the difference between a motif and the domain. Then we will discuss the difference between MES and NES. And then last thing, we will discuss the steroid signaling pathways. So brief introduction, I will go through them very fast because there's nothing really to explain. So basically, what is a hormone? Hormone is a chemical messenger that will transport the signal from one cell to another. Four major chemical classes of hormones are, we have steroid hormones, peptide hormones, amino acid, and prostaglandins. In this lecture, we will discuss the steroid hormones. The function of steroid hormones and some examples. We have, first of all, glucocorticoid. An example is a cortisol, which serves as a carbohydrate regulation. Mineralocorticoid, such as mineral balance. Reproductive functions, examples are gonadal steroids like androgens and estrogens. Another functions like inflammation, they are included or they play a role in the inflammatory responses, stress, bone metabolism, and so on. So what are types of steroid hormones and examples? Glucocorticoids, example is cortisol, which is the which is a representative hormone. It's important in sugar regulation in our body. Mineralocorticoids, example is aldosterone, which the main function of aldosterone is to retain salt, to maintain a balance between water and salt in our body. So that's the main function of aldosterone. We have androgens like testosterone, estrogens like estradiol and esterone, and progestogens. Before I start the main topic of this lecture, I want to give you an overview of what I'm going to discuss today, so you know what I'm talking about. In this lecture, we are going to discuss the steroid hormone signaling. So let's say we have a steroid hormone in the blood plasma. Usually, or not usually, always they are bound to a plasma protein, or sorry, protein carrier in the blood. Once they reach the cell, before, let me orient you here. So this is the blood, this is the plasma membrane, this is the cytoplasm of the cell, and this is the nucleus. But let's say the steroid hormone reached the cell, and so because this is the steroid hormone, once it reaches the cell, it will penetrate the plasma membrane because it is a hydrophobic. Remember that all steroids are hydrophobic, and so they have the ability to enter the cell membrane or to penetrate the cell membrane. So once they penetrate, they will bind to their receptors, and once they form the hormone receptor complex, they will introduce transcription. So this is the general idea of this lecture. Steroid hormone entering the cell, binding to its receptor. We have multiple types of receptors. We will discuss them, and then enter to the nucleus, bind to the DNA, and then induce transcription to produce or synthesize a certain types of proteins. This region here is called the hormone binding site. And once this receptor binds to the DNA, this will be called the DNA binding domain, which is the zinc finger protein, which is very important, and you have to know it. So steroids are hydrophobic. They can cross the plasma membrane. Okay, they bind to the receptors. They move to the nucleus. They bind to the DNA, induce transcription. And you have to know the DNA binding domain and the hormone binding domain, which we will discuss them. First of all, let's talk about the steroid hormone receptor. So this is the receptor. Remember that the receptors are proteins, so that's why we have an N-terminal here and a C-terminal. The steroid hormone receptor contains multiple domains. Number one, we have variable domain. Variable domain basically interacts with other transcription factors. Again, what are transcription factors? They are molecules that induce or suppress the transcription. Number two, we have the DNA binding domain, which is the zinc finger, which is very important, and you have to know You have to know it. طيب. The DNA binding domain. So this region here, once it enters the nucleus, it will bind to the DNA through this region, the DNA binding domain. Type 3 is the domain for dimerization, and I will explain what dimerization means. And number 4 is the hormone binding domain. So when the hormone comes from the blood and enters the cell, it will bind to its receptor through this region, number 4. Once it is bound, and once we have a hormone receptor complex, it will enter the nucleus, bind to the DNA through number 3. Oh, sorry, number 2, which is the DNA binding domain, the zinc finger motif. So this is the steroid receptor, those two. We have multiple steroid hormones, glucocorticoids, mineralocorticoid, progesterone, androgens, estrogens, and so on. So this green triangle is the steroid hormone. It will bind to its respective receptor. In this case, it is cortisol, so it will bind to the glucocorticoid receptor. Once it binds to its receptor, an important concept of steroid receptors is dimerization. So once a hormone binds to its receptor, it will undergo dimerization. As you can see here, the yellow thing, this is called dimerization. And dimerization is important for the proper function of steroid receptors. Another thing, we have the DNA binding domain here. So this is the DNA sequence. Once the hormone binds to its receptor, it will bind to the DNA. So this is the DNA binding domain, which is number 2. Yeah, now the function or the difference between a motif and a domain. I will highlight the key points you have to know about the motif and the domain. First of all, they are both functional units of proteins, so they serve a certain function. Let's start with the motif. What you have to know about the motif, first of all, it is an amino acid sequence. It can be found in different domains of a protein. They have or they contain 2 or more secondary structures. They are connected by a loop or a turn, and they are considered as super secondary structure. Please know that a motif is a super secondary structure. That's for the motifs. The domain is an independent folding unit. It's more complex than a motif in terms of their structure. So I highlighted the key points you have to know for the motif and for the domain. For the motif, so it's simpler than the domain in terms of structure. They are considered a super secondary structure, and this is important because they have 2 or more secondary structures and they are connected by a loop or a turn, and they can be found in different domains of a protein. What does number 3 means? And know that a motif can be found in different domains. Like here, we have multiple domains. Motif can be present in number 1, 2, 3, or 4. Type. For the domain, just know it's an independent folding unit, and it is the next higher level of organization than the motif. That's it for the motif and domain. So now let's discuss in details the structure of a hormone receptor, a steroid hormone receptor. So first of all, it's a protein. So it contains an N-terminal and a C-terminal. We have a hormone binding site. So once the hormone enters the cell, it will bind to the receptor through this site, which is hormone binding site. The other important thing is the DNA binding domain. Now this DNA binding domain, once this receptor enters the nucleus, it will bind to the DNA, because we said the general idea of this whole lecture is that we want the receptor hormone complex to enter DNA and induce transcription. So that's why on the receptor, we have a domain called DNA binding domain. It will go to the nucleus in order to bind to the DNA. So what are the functions of this DNA binding domain? First of all, obviously bind to DNA, but how? It's through the zinc finger motif. So please know, this is a high yield information. You have to know that recognition of DNA sequence is by the zinc finger motif. So the DNA binding domain contains zinc finger motif that will recognize DNA in order to have this binding between the receptor and the DNA. Another thing here, the NLS, the nuclear localization signal, basically this molecule, once we have this receptor synthesized, newly synthesized by a ribosome, it will, this NLS will tell this receptor to go to a certain place. For example, to go to a nucleus. And that's it for the NLS. طيب. Now let's classify steroid receptors. We have 2 types. our two classifications. We have a MES and the NES. MES is the membrane-initiated steroid receptors. The NES is the nuclear-initiated steroid signaling, sorry, not receptor, signaling, steroid signaling. And it's so important to know the difference between them. We will discuss them. But for the steroid membrane receptors, from its name, the steroid receptor is found on the membrane of the cell. So it's not inside, it's not in the cytoplasm, it's not in the nucleus, it is only present at the membrane. The other let's say pathway as or classification of the receptors is the intracellular. Now the intracellular is considered as the NES, or the nuclear initiated. From its name, intracellular, so it's either or let's say that this category is subclassified into two types, type 1 and type 2. Type 1 is present in the cytoplasm, type 2 is in the nucleus. For type 1, it's mostly combined with heat shock proteins, and you have to know the examples of hormones that act by type 1 receptors, or act on type 1 receptors. Examples are 6 steroids like androgens, estrogens, and glucocorticoids like cortisol. And those form homodimers only. Now the concept of homodimers and heterodimers I will discuss in the next slide. Type 2 is only present in the nucleus. It's often bound to DNA, which makes sense because if the receptors are in the nucleus, it's mostly bound to the DNA. And you have to know the examples. We have thyroid hormone and vitamin D. So what does that mean? Yani when thyroid hormones enter the cell, they will directly go to the nucleus in order to bind to the receptors, which are type 2 steroid intracellular receptors. On the other hand, if a 6 steroid, let's say androgen or testosterone to be more specific, once it enters the cell, it will go to the cytoplasm and not the nucleus in order to bind to type 1 steroid intracellular receptors. Both type 1 and type 2 are considered as NES. Now type 2 can form homo and heterodimers, which I will discuss next. One more thing about the estrogen receptors, that they are considered as nuclear receptors located either in the cytoplasm or in the nucleus. What does that mean? So basically you have to know that the estrogens are a class of nuclear receptors. What they mean here, they are located in the cytoplasm. Here it is located in the cytoplasm, but eventually when the estrogen comes, it will eventually end in the nucleus. And so you have to know that they are nuclear receptors. For the estrogen receptor type alpha is found predominantly in the nucleus, both in hormone stimulated and untreated cells. So irrespective from the binding of estrogen to its receptor in the nucleus, what does that mean? If the estrogen is present or not present, either ways, the estrogen receptor alpha, type alpha, will be present in the nucleus. So if there is a hormone or there is not a hormone, doesn't matter. This type of receptor, alpha, it will always be, or mostly be in the nucleus. Now this slide is just to explain the concept of homodimerization and heterodimerization. So let's take an example for this one. So we have a cortisol. It will bind to its receptor. It will undergo dimerization. You have 2 receptors. So when you have 2 receptors of the same type, and when the hormone binds to those 2 receptors, they undergo dimerization. And because they are the same type of receptor, they undergo homodimerization, because homo means same. And so when you have 2 types of, or 2 receptors of the same type, it is considered homodimerization. This is the same thing. It's homo because you have RXR, so the same, or the retinoid X receptor. They have, or 2 receptors of the same type. On the other hand, the heterodimers, we have it when we have 2 different receptors. When the hormone binds to a receptor, and then, or basically when you have 2 different receptors dimerizing, it's called heterodimerization. An example here is thyroid hormone or vitamin D3. As you can see here, again, thyroid hormone, vitamin D, they can form both homo and hetero. So in summary, homo, 2 receptors of the same type, hetero, dimerization, is when we have different receptors. Yeah, just when we have a different receptors bound to a thyroid hormone or vitamin D. And that's it for the homo and heterodimerization. طيب. Let's further classify the steroid receptors, the intracellular receptors. We have type 1 and type 2. Let's talk about each one of them and how each one works. Remember that type 1 is nuclear, right? Nuclear or cytoplasmic? It's cytoplasmic. So type 1 is cytoplasmic. So it's present only in the cytoplasm. Examples, again, 6 hormones, glucocorticoid and mineralocorticoid. So when the hormone in the bloodstream enters the cell, it will bind to its receptor. So now we have an active receptor complex. This complex will enter the nucleus, and then it will bind to DNA. To be more specific, it binds to a region called hormone response element. Of course, as homodimers, because we said type 1 forms homodimers. Now, when this complex binds to the hormone response element, it will expose their gene regulatory site, and now transcription factors can act on, and therefore we have induction of transcription. And this is basically how type 1 works. So remember, it's cytoplasmic. So the hormone will bind to the receptor on, or let's say in the cytoplasm, and then there is translocation. Translocation to where? To the nucleus. Why? Because we need to bind to the DNA on the hormone response element. Type 2, on the other hand, which is the nuclear receptor, we have the, when it is unbound receptors, what does unbound means? Meaning there is no hormone. The hormone didn't come. The receptor, it's on its own in the nucleus. So once there's unbound receptors, we need something to inhibit transcription because there is no hormone. There is nothing telling us to induce transcription. And because this type 2 receptors is already in the nucleus bound to DNA, we need something to inhibit transcription. And this thing is called the co-repressor proteins. So when we have an unbound receptors, they are complex in the nucleus with co-repressor proteins. And this co-repressor protein will inhibit transcription. Why? Because again, we don't have a hormone. We don't have a steroid hormone or vitamin A, vitamin D molecules. However, when a steroid hormone comes to the nucleus, it will bind to the receptor. And this receptor then will release the co-repressor protein. So no more co-repressor protein. And therefore, the co-activator proteins now will come into play. And then we will have an initiation of transcription. Again, examples are like thyroid hormones and vitamin D. So this is the only difference, that in type 2 receptors, which are in the nucleus, we have something called co-repressor proteins. These co-repressor proteins are present when there is an absence of a hormone, of a steroid hormone, like vitamin A or vitamin D, in order to inhibit transcription. However, on the other hand, when we have when the hormone comes into the nucleus, it will bind to the receptor. Then the co-repressor proteins will be released. The co-activator proteins will come into play and then initiate transcription. Let's talk about the steroid hormones signaling pathways on the molecular picture type. So this is the blood vessel. This is the bloodstream. We have a cell here. This is the cytoplasm, and this is the nucleus. So let's start by here, number 1. Type. The steroid hormones, they are always in the bloodstream bound to a protein carrier. Once they reach the cell they need to go to, they are released, and then they can penetrate the plasma membrane. Now for the steroid hormone signaling, we have 2 pathways. We have a minor pathway, which is this one, in which the steroid hormone comes and acts on or bind to a cell surface receptor. So this receptor is only found on the membrane of the cell. So once the steroid hormone binds here, it will activate or induce, I would say, the rapid response. So this binding will activate a cascade of signals or it will activate or increase the activity of second messengers like calcium, like cyclic AMP, in order to produce a rapid response. And rapid here is a keyword for this pathway. And this is what we refer as to the MES, or the membrane initiated, in which it induces a rapid response. The other pathway, which is this one, this whole thing is called the NISS, or NISS, the nuclear initiated. And we said that the intracellular receptors, we have two types, the cytoplasmic and the nuclear. So the NISS includes both cytoplasmic and the nuclear. What's the difference between them? So once the steroid hormone binds to the cytoplasmic receptor, it would translocate into the nucleus, or the other one will wait for the steroid hormone to directly come from the bloodstream to the nucleus and bind here. Eventually, both of them will bind to the DNA through the DNA binding domain. Then we have transcription activating, and then we have translation. Therefore, we can have a new protein, and this is the whole idea of the steroid hormones. So you have to know the difference between MES and NIS. Now, NIS takes time. However, the MES is the rapid response. So this pathway, it exerts its effect or produce a response very quickly, fast. However, this pathway takes time because there is production or synthesizing new proteins, and again, gene transcription, translation, blah, 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 takes a lot of time. Type. Overview of steroid signaling. Again, we said for the steroid signaling, we have two pathways. We have the classical signaling, which is the NIS, the nuclear initiated steroid signaling. And the main function of this pathway is gene transcription, which will then eventually allow for protein synthesis. And this may take minutes, hours, or days. So it takes a long time to be done. Now, the rapid signaling, or the MES, the membrane initiated steroid signaling, it exerts its effect or produce a biological response very quickly. How? By either the general idea of MES, that it's increase in second messengers. Examples are increasing intracellular calcium, activation of G proteins, and then activating a cyclic AMP, which is a second messenger in this case, and also protein kinases. And then both of them, they are important for the normal function of steroid hormones. So the main difference between NIS and MES, so this one is the NIS, the nuclear initiated steroid signaling. Once the hormone binds to the intracellular receptor, intracellular, either cytoplasmic or nuclear, they will interact with the genome. So they will bind to the DNA, leading to protein synthesis. And this may take an hour to produce a specific response. So this is the nuclear initiated, this is the NIS, nuclear initiated steroid signaling. This one is the MES, the membrane initiated. So once the hormone binds to the membrane receptor, it will increase second messengers like calcium, cyclic AMP, or activate also kinase activity. They will produce a response in seconds. And this is the main difference between MES and NIS. Okay. Checkpoint. Is there any questions? تمام. طيب, let's move on. Now let's talk more about the NIS and the MES. So in the absence of hormones, we have estrogen and progesterone receptors. They are nuclear receptors. We talked about them, that estrogen is a nuclear. We have type 2 receptors, which are also nuclear receptors. And remember that type 1 is cytoplasmic. Examples like glucocorticoids and androgens. So you have to know the difference between type 1 and type 2. Now, in the presence of hormones, I will demonstrate on the diagram. So here we have the steroid hormone in the blood. It will penetrate the plasma membrane. It will enter the cytosol, or the cytoplasm. It will bind to its respective receptor. And then we have this complex. This complex is called the hormone receptor complex. Now it will go to the nucleus, and then bind to something called hormone response element. This one is called the hormone response element. Once this complex is bound to this hormone response element, now we have an induction of transcription, and then we can make mRNA, and then we can make a new protein. So steroid hormone will stimulate the receptor. They will bind here, so the hormone will bind to its receptor. It will enable it to translocate to the nucleus here, and the nucleus, it will bind to something called hormone response element, this one, on the enhancer region, and then activate gene transcription. Now about the MES, remember that MES is okay, so it's the membrane initiated. So the receptor is found on the plasma membrane and not intracellularly. So once we have the steroid hormone binding to this receptor, it will activate certain protein, certain signaling proteins. Examples are G proteins, growth factors, tyrosine kinase, SERC, and so on. So once those are activated, they will activate kinases, including protein kinase A, C, phospholipase C, MAP kinase, and so on. So once those are activated, they will phosphorylate targeted proteins or molecules that will induce a rapid response. And this is the whole idea of MES. We have a steroid hormone binding to the receptor on the plasma membrane, activating signaling proteins like G protein, growth factors, and so on, that will lead to the activation of kinases, and those kinases will phosphorylate molecules that will induce a rapid response. Just know that here that the steroid hormones bind to the MES, or the membrane-initiated, or the receptor on the membrane at the cavula, because there is a past question about this cavula. The fact that MES induce effects faster than the NES is because they activate an already existing proteins, unlike the NES, in which we have to induce transcription, then make mRNA, then make translation, and then make proteins. No. So that's why NES takes longer time than MES. Because in the MES, we don't require to synthesize new proteins. Again, an example of this is EGFR. So EGFR is implicated in MES, and its activation can result in the sustained MAP kinase signaling. So let's say this is a EGFR, it will activate the MAP kinase, and the MAP kinase will phosphorylate a certain molecule that will induce a certain rapid response. We have crosstalk between MES and NES in order to evoke biological responses. Right. The last concept, we have the hormone specificity, which basically means that for each steroid hormone, we have a specific receptor that binds to. Let's take an example of cortisol. Cortisol here will bind to its receptor, a specific receptor called the glucocorticoid receptor. So once we have this complex, and of course it forms a dimer, so they undergo dimerization as we mentioned, then it will enter the DNA, it will bind to the HRE in this case, or not HRE, let's say the hormone response element, right, on the DNA sequence. In this example, it is called the glucocorticoid response element. So once the complex binds to this GRE, it will induce transcription. And this is the whole thing of, this is the whole idea of this slide. That we have hormone specificity, example, cortisol has a specific receptor in which it binds to. So again, activated receptors are specific to certain hormone response element. In this case, it's GRE, or glucocorticoid response or receptor element, which are specific for only those receptors. This GRE may be located upstream, downstream, and can regulate their expression from distance. Type. Last slide, signal response time. Again, the ion channel receptors are measured in milliseconds, so they are very fast. For GPCRs, the G protein coupled receptors, transmembrane enzyme receptors, and JAK-STAT, they take seconds to minutes, and this is the MISS, the MISS, membrane initiated. And for the steroid receptors, which are mainly NES, this classification or this category is mainly the NES, the nuclear initiated. It takes a long time, hours or days. This is the end, and I will start practice questions really quick. First question. DNA recognition is done by which of the following in a steroid receptor? DNA binding domain, hormone binding, zinc finger motif, or hinge region? Let me take the pen. So we are saying what the recognition. Recognition is done by a zinc finger motif, and this is a high yield information you have to know. Now, DNA binding domain, it binds to DNA. However, the one that recognizes the DNA is the zinc finger motif. And remember that the DNA binding domain contains zinc finger motif, and this zinc finger will recognize the DNA and binds to. Type. A particular cell type under study is known to be the target of a certain hormone's action. The hormone has an intracellular receptor, so intracellular receptor, that normally resides within the cell's nucleus. Okay, so this is in the nucleus. The most likely identity of the hormone is it insulin, growth hormone, estrogen, adrenocorticotropic hormone, prolactin? So mainly they are saying here intracellular, first of all. So they are giving a clue that this is a steroid hormone. So insulin is a protein, so it's not the answer. Growth hormone also is a protein. So the only steroid here is estrogen, so the answer is C. Which of the following regions within an androgen receptor protein contains zinc finger motif? Again, we said that zinc finger motif is contained in the DNA binding domain of the steroid receptor. A steroid-responsive tumor cell has abnormally increased activity of MAP kinase signaling. Okay, so what comes to your mind here when you read MAP kinase? Is it MES or NES? When you hear or when you read MAP kinase, okay, so now you know it's a MES and not NES, so it's membrane initiated. Remember the rapid response one. Which of the following aspects of steroids signaling may be involved in this abnormally sustained signaling? So no need to read all this. So just you know that this is a MAP kinase signaling. Okay, now you know it's a MES. So on MES, you will choose the membrane initiated. And that's it for this question. Tamoxifen, which is a drug used to treat breast cancer, is a competitive inhibitor of estrogen receptor. Okay. So once you give this drug, the estrogen receptor in the nucleus will be inhibited. Type a zinc finger protein. Tamoxifen will therefore affect gene expression in these cells mainly by how will it affect the gene expression? So when you give this drug, they will go to the nucleus, they will inhibit the estrogen receptor. So once this is inhibited, even if the estrogen came to the nucleus, it will not bind to its receptor, and therefore you will not have there will be no induction of transcription. So the individual will suffer from preventing transcription of estrogen responsive genes. Why? Because the drug inhibited the receptor, so the hormone estrogen cannot bind to its receptor, therefore there will be no induction of transcription. Last slide. In the inactivated state, type 2 steroid receptors are attached to, so we have inactivated state, first of all, and then type 2. We said that the type 2 are nuclear, right? Nuclear, so they are in the nucleus type. So we said the receptors that are present in the nucleus, we need something to repress them or inhibit them from initiating transcription when there is an absence of the hormone. And because they said it's in the inactivated state, so the answer is the repressors. Because when there is no hormone, the repressors will inhibit the steroid receptor, and therefore we don't have an induction of transcription. However, when the steroid receptor comes to the nucleus, binds to the receptor, the repressors will be released, and then the coactivators will come into play to induce transcription. Last question, the membrane-initiated steroid signaling, MISS. Basically, what activates receptors at the yeah, this is the one. I told you about the cavula. Remember this word. The DMS activates the receptors at the cavula. This one. This one. Okay, so the MES act here. At the, I think it's called the cavula. Okay, whatever. And that's it. This is the end. Does anyone have any questions? Okay, thank you for listening and see you. Assalamu alaikum wa rahmatullahi wa barakatuh.